Hello, in this video we're going to talk about cell membranes and transport. So here we have a picture of a cell membrane, sometimes called the fluid mosaic because it looks like a mosaic with all these different pieces and it is very fluidy. So let's have a look at the phospholipid bilayer. Remember the phospholipids, these guys are polar. They've got charge on them right there. Charge. These guys are nonpolar. This is kind of sloppy. Nonpolar. So when we put them together, the nonpolar regions are going to want to stay together. And then the polar are going to be on the inside and outside around the polar aqueous internal and external part of the cell. So it forms that lipid bilayer. Okay, so these phospholipids kind of float together and there's some movement back and forth. So things like oxygen and carbon dioxide can pass through there pretty easily. Larger molecules can't, and also polar molecules generally cannot because of the nonpolar internal lipids in there. Okay, another thing about the makeup of the phospholipid bilayer is if you've got a lot of saturated fatty acids in there, they tend to lie pretty straight. They're linear and they pack together really tight. So that's going to be more viscous or like a thicker membrane. If you have a lot of unsaturated fatty acids in the membrane, they're kinky. All those double bonds cause them to bend and they don't pack together very well. So that's going to be kind of like a more flexible, a more fluid membrane. That's going to be more useful in something like a cold water fish, where you don't want your membranes kind of stiff and inflexible. You want to have more fluidity in there. So the type of membrane, warm-blooded animals will typically have saturated fatty acids in their membranes, whereas cold water animals oftentimes will have unsaturated, or like a caribou that's got in its hooves, where the membranes get very cold, would have more unsaturated fatty acids to give the membrane more flexibility in those cold temperatures. And then cholesterol is an important molecule. Remember, it's a ringed lipid, and what it does is it kind of binds in there and kind of holds together some of those fatty acid tails and gives it some structure but also flexibility and makes that membrane kind of reinforced and gives it a little added strength. So those are some parts of it. So other parts of the phospholipid bilayer or the fluid mosaic of the membrane are these proteins, right? And the proteins have several functions. We'll talk about those in a second. And then we have the inside and outside of the membrane. So let's look at some of the functions of these membrane proteins. First off, a lot of them move things in and out. So we have either A is a channel protein, opening up a channel. B is a protein that's moving something more actively using energy, called active transport. So proteins are involved in transport. Some proteins on membranes, especially internal membranes like the mitochondria and the endoplasmic reticulum, are enzymatic. So embedded proteins can also be enzymes and serve enzymatic processes. On the cell membrane, proteins play an important role in terms of receiving a chemical signal, a hormone, and sending that signal into the cell, initiating a response. That's called signal transduction, receiving a chemical signal and causing a response. Other proteins over here on the right just can bind to and join with other cells to kind of form a tissue layer. This is a sugar attached to a protein, and that's called a glycoprotein. This would be another cell, perhaps a white blood cell, that is sort of checking that glycoprotein. So that allows our cells to recognize cells as cell-to-cell recognition. That's important in terms of our immune system, knowing if cells are friendly or foreign. And then this is a protein binding to the extracellular matrix and also the internal cytoskeleton of a cell. So again, holding the cell in place is an important role of those, attaching to the cytoskeleton inside and the extracellular matrix outside of the cell. All right, so movement in and out of cells. So we know that diffusion is just the movement of particles, and we know that some particles like oxygen and carbon dioxide can move across the membrane, and we call that movement diffusion. So if we have some molecules and they can pass through this membrane, over time, due to their random movement, they're going to diffuse and eventually reach equilibrium, equal concentrations on both sides. And if we have two different solutes on and they can both pass through the membrane, they'll both diffuse and again reach equilibrium, and it'll look something like that slide on the right. Okay, well, this is important because some molecules can move through membranes, other molecules cannot. So I want to introduce a term, and water can pass through membranes, and water, if it is in a concentration that has less dissolved particles, hypo means below, tonic is like dissolved particles, or you can think about tonic as just a solution. So hypo means less dissolved particles in that environment. Hyper means above, so this means there's more dissolved particles in that solution. So if it's a hypertonic solution, there's more dissolved stuff in the water, hypo less. Okay, so let's look at some examples of that. This relates to osmosis, because water is always going to move from a concentration of greater water concentration to less water concentration. And the movement of water across membranes is called osmosis. So there you go, it's reached equilibrium. How does this affect cells? So if we take a red blood cell and we put it in a fresh water solution, again, that's called a hypotonic solution, less dissolved particles in that fresh water. That means water is going to move from higher to lower concentration. From that fresh water into the cell is going to swell up and eventually explode or lyse, break apart. If it's a plant cell, it will move in, water will move into the cell, but the cell wall will prevent it from swelling up and exploding, and it will just be called turgid. It's pressurized, right? It's going to, the pressure inside the cell from the cell membrane pushing back is going to reach an equilibrium, okay? Isotonic, iso means the same. So if we have the same concentration of dissolved particles inside and outside the cell, water moves in and out, but it moves in and out at about the same rate. So our cell's in this nice sort of homeostatic balance, okay? And same thing in a plant cell, water moving in and out at about the same concentration. We have a hypertonic solution. That would be like a saltwater solution. We've got more dissolved stuff out here than inside the cell, and we're going to have water go from higher to lower concentration, moving out of the cell. Okay, that's going to cause the cell to shrivel. And if you take a plant cell and put it in a saltwater solution and look at it in the microscope, you can see the membrane is all shriveled up as water moves out and the membrane shrivels up. Okay. So here's an example of a paramecium. Paramecium live in fresh water. So they've got this problem. Water is constantly diffusing from fresh water into the cell, right? And they're going to swell up. Well, they have this thing called a contractile vacuole. It's like a bladder for them. They're going to continue to move that into their contractile vacuole and fill it up, just like if you drink a lot of water, your bladder fills up with urine, right? And then what do? They go to the bathroom, they contract it, and this is showing it contracting, squeezing all that water out. So paramecium has this mechanism, this adaptation to avoid sort of swelling up and exploding due to this osmotic pressure, water moving in. They fill up this contractile vacuole and they contract it and they don't rupture. All right, so again, movement across membranes. So any particles like oxygen, carbon dioxide that can pass through that phospholipid bilayer can move into and out of cells by a process called simple diffusion, okay? Other particles like salt ions can't pass through that membrane, but we have proteins. In this case, there's a channel protein that opens up a channel and can allow them to pass through. Because the protein is helping the diffusion, we can say it's helping or facilitating the diffusion. So we call that process facilitated diffusion. Protein is helping something go from high to low concentration. These are both passive processes, right? There's no energy required. That's the way things want to move due to their random movement. So we call those passive transport. If we need to move something from lower concentration to higher concentration, we need to go against that concentration gradient. So we need to use energy, the energy molecule ATP, and that process is called active transport. Okay, so let's look at some examples. So here's two examples of facilitated diffusion. The first on the left is a channel protein. It's just opening up a channel and it's allowing particles to move through the membrane. So that's just a channel protein. On the right, it's a little bit more specific. This is actually binding to a specific solute and moving it across the membrane. So this is what we call a carrier protein. It's carrying a specific solute across the membrane. But neither of these processes are requiring energy. They're moving something from high to low concentration. Okay, here is a neuron, and these proteins on the neuron are called the sodium-potassium pump, and it's doing what it sounds like it's doing. It's pumping sodium out of the neuron and potassium into the neuron to establish this concentration gradient. And to do that requires energy, right? You're moving something from lower to higher concentration, and that's an active process. ATP is involved. There's your ATP, and that takes energy. So this is an example, the sodium-potassium pump, of active transport to sort of recharge our neurons so that they can fire. They can send a nerve signal, and we'll talk about that in a couple months. Here's another example. So in cells, we have this thing called a proton pump, and it can pump hydrogen ions across a membrane. Remember, the mitochondria does this with the electron transport chain. It uses that electron transport energy to pump hydrogen ions in that intermembrane space. Here we have that same process undergoing active transport, moving that hydrogen ion. And then why would a cell want to do that? Well, it can use that hydrogen ion concentration then to pump back through a co-transporter and link up with sucrose and move sucrose into a cell. This process is called co-transport and involves the active transport of hydrogen ions to then allow the co-transport of sucrose into cells so cells can use sucrose for energy. Okay. And then the last three ways that we can get things into and out of cells, let's say you have something really large you need to get into a cell, right? Like this bacteria is getting engulfed by an amoeba, right? Well, a bacteria can't just be moved by proteins or by simple diffusion. You got to wrap your entire membrane around it. And that process is called endocytosis, right? Entering the cell, cytosis is just the cell entering the cell by wrapping the membrane around it. And if it's something solid like a bacteria or some food particles, we call that phagocytosis. Just remember, phag, it's the F sound like for food, okay? If we're taking dissolved particles, liquids into a cell, that's called pinocytosis. So taking in of liquids is pinocytosis. And then what happens occasionally is there's a signal out here or some kind of hormone or chemical signal that binds to a receptor, and that initiates sort of an endocytosis process. The membrane then wraps around that hormone or that protein and takes it in, and that's called receptor-mediated endocytosis. So this is, again, moving things through the membrane by having the membrane wrap around them, and that's the last type of cell transport. And that's it for this video.